talk about the aseptic techniques. Okay, aseptic techniques is a very, very important technique to assure that uh, your culture will not be contaminated by the microbes. Okay, so contaminants is the main hazard in tissue culture, as all of us know. So um, I believe all of the people that have gone through the process of tissue culture, be it the plant tissue culture or animal tissue culture, they must have facing some of the contamination issue. So this is something that we will try to eliminate as possible, right? And cell growth in vitro are not protected by the in vivo immune system because you excise a piece of the tissue from the organism. And once you excise it or you isolate it from the animals, these chunks of tissue were not protected by the immune system of the organism. So it is very fragile. And you need to culture them in an artificial environment with the medium. And so it is extremely important to provide sterile environments with the right amounts of nutrients and also other parameters to make sure that the cells continue to survive and replicate. And the common contaminants found in the tissue culture are such as bacteria, viruses, mycoplasma, fungi, and yeast. These are the 5 main contaminants that are commonly found in a tissue culture. So what are the potential source of contaminants? First is the operator. You are the operator. Students, you are the operators. So when you conduct your experiment, you need to make sure that you are clean enough. Okay, for example, for ladies, if you have the long hair, you need to tie it out and put a cap on. Because the long hair, if you don't tie it up, yeah, they may trap dust. Okay, when you work from your dormitory, okay, all your the dust will be trapped on your hair, and once you move your hair, then all the contaminants, all the dust, you know, will be get into the air, and at the end they may drop down on your media and lead to the contaminations. Okay, the unclean work surface. Some people they are I would say some of the students are quite irresponsible. So after they use the surface or after they use certain area, they don't clean up. Okay, they don't clean up, and this will lead to the contaminations for the people who come next. Another one is the atmosphere. Okay, in the lab, because sometimes the lab can be quite dusty. Yeah, especially like in Unimas, we are very funny that some of the lab they are naked on top of the ceiling, and you know when the ceiling is have you been to any of our lab before? Do you have a chance to go visit any lab?
No, doctor.
No.
No,
So,
yet.
so actually our lab have no ceiling. So can you imagine all the dust will be trapped in the air, you know, they may drop down right into your media. So this is the environment. So that's why you need to make sure that the pressure is correct, and then you need to filter it with the HEPA filter to make sure that the air in the tissue culture room is clean. And in the improper sterile glassware and plasticware, sometimes if you don't sterile them with the correct methods, okay, this glassware and plasticware can get contaminated. And if, for example, if you wash them with the dish detergent, yeah, the cleaning of the washer, and the detergent may trap if you do not rinse it properly enough with the water, and they may, you know, they may stick on the glassware or the plasticware, and when you make your own media, this detergent can get into your media and contaminate your media, right? And the other one is the media. Media, if you don't filter them, then you have a high chance to get contaminations as well. And if you do not have the proper procedure to, you know, make your own media, and you know, media can be easily be contaminated because media are full of nutrients. So it is the bacteria, the viruses, the microplastic yeast, or okay, they like it so much because they're full of nutrients. Yeah, so it is very important to make sure that the media is extremely clean, or I would say sterile, that's to avoid any contaminants. All right. So how to observe contaminations in a tissue culture? You can use your naked eye. Look at the turbidity of the media. So the bacterial growth or the highly acidic due to the media exhaustions. For example, before you use your media should be clear. It should be turbid. It shouldn't be turbid, right? Because if it is turbid, that means something grow on it. So you are not supposed to grow your cell line if your media already turbid before you culture your cell line. Does it make sense?
Yes.
Yes, doctor.
Do you know what is turbidity?
It's like cloudy.
Yeah, it's cloudy, like sudah keruh. That means ada pertumbuhan di dalam media itu. Okay? So supposedly before you grow any cell line, your media should be very clear, right?
Yeah.
Yes, doctor.
Yeah. Okay. And the other way is you can observe them under a microscope. You can see the Brownian movements of the bacteria, the granulations or degradations of the cell in the viral or mycoplasmic infections, the mycelia in the fungal infections, or string of small bits due to the yeast infections. You can look at them under the microscope. That's why you have the microscope room in your tissue culture lab. Okay, it is very important to have a microscope to observe your culture from time to time to avoid any contaminations. Okay, if you found any contamination, then you have to stop the experiments and you probably have to change your media. Right? Then what are the precautions of maintaining the sterility? First is the cleanliness of the operators. You yourself need to be very clean. The tissue culture room also needs to be clean. This is very general. Okay, like workbench, you're working with the culture, your equipment have to be clean, handling with the open culture, handling with the microwave plate and cross-contamination. These are very, very common, these are general knowledge. But to make sure that these precautions are well taken care of is a challenge, because the tissue culture room are not being used by you yourself. You may have share the facility with other students and you don't know their habits. Yeah, you don't know their habits, and that is why, okay, the precaution step have to be really practiced well in the tissue culture lab to avoid any contamination. Once your cell line contaminated, that's been there is a wastage of the resources and also time of doing experiment. Okay, how much time can you afford to let your cell contaminated because you are chasing after the deadline and your schedule, right? Then cross-culture contamination is the most difficult types of contamination to detect, because especially when the cell line morphology is very similar, it is very, very difficult to detect them. Yeah, and it can be carried for years without detections and can be carried as mixed culture for a long period of time. And once you have these contaminations, the result that you obtain from the experiments is no longer valid. Okay, because what we want is a pure culture cell line. The result from the pure culture cell line, not the cross-culture contamination cell line. Okay, and the contaminating cell might take over the entire culture, causing the loss of the original cell line that you have culture and generate, that you generate many years ago. So you need to very, very careful to avoid cross-culture contaminations. Okay, because I have encountered this before, and it is very difficult. Yeah, because they may give you the false result. Okay, and it is reported that about 30% of the data that's published in the paper, the results were from the cross-culture contaminations. So it is extremely important, and you have to practice extremely careful SOP and also for your tissue culture. Okay, When a new cell line is established in the laboratory or is brought into the laboratory from another laboratory, excuse me, commercial or research institution, it should be tied to determine identity. Because if you don't do that, okay, if you don't type, if you don't identify the identity, so it is impossible for you to proceed to the next step. So these typing methods may include the isozyme typing, the karyotyping, like looking at the number of the chromosome, and also the cell identity determination by looking at the proteins, okay, the enzyme, right? So it is very important once you get the cell line, you know, from any other sources, the first thing that you need to do is do the typing, okay? And so overt cross-contaminations, it is very important to do not have the two cell lines in a hood at the same time. Because humans tend to be very careless sometimes, and you don't know this cell line can be contaminated because of your carelessness, and do not ever put the same pipette tips or anything else into two sequential stock culture. Okay, this is a common mistake done by the students, that you don't change your pipette tips. Okay, for example, you just pipette the cell line A, and after that you don't change the pipette, and then you put it in the cell line B, and this cell line from A will contaminate the cell line B, and that is what we call the cross contamination. Okay, and number 3 is wipe down. Okay, wipe down the hood surface and wash hands, and change your glove in between the handling different cell line. Don't save the glove. If you need to change, you need to change it. Okay, especially if you're handling different cell line, because the cell line may stick on your glove, and this may get into your other cell line. So be very extremely careful when you're handling them. Or store a frozen cell line in the vapor phase of the liquid nutrients, or use glass freezing valves and leak test. You need to check, make sure that there is no leakage. Okay, once there is a leakage, then this cell line can contaminate the other cell lines as well. So be very, very careful and use the correct vials, which can withstand some minus 70 degrees Celsius. Okay. So far, can you follow me?
Yes, doctor.
Yes, doctor.
Yes, doctor.
It's quite general. It's very easy to understand, but to practice it is a challenge. Okay, so the tips to reduce contaminations: the benchtop, the hood surfaces, the microscopes, and other work surface should be wiped down periodically with 70% ethanol or 75% ethanol, or other germicide. Okay, and the inside of the refrigerator should be kept clean and unclustered. Okay, but a lot of time, if you go to the refrigerator of most of the lab, it is like very messy. Not to mention the lab, you look at your home, you look at your own refrigerator. Is it clean? Or you just chuck everything inside your refrigerator? Or your mom probably just chuck everything inside the refrigerator. How frequent you clean your refrigerator at home?
Once a year.
Once a year, maybe during Chinese New Year or Raya or Christmas. Or probably you have your Christmas cake freezing somewhere in the fridge by your mom. Because I know most of the mom, they think a refrigerator is something like a magic box. They can just put anything inside there and make sure that it's fresh. I don't know about your mom, but my mom has been trained. I have trained her not to put anything inside the refrigerator. If you want to put in the refrigerator, you have to put in the proper box that are provided by us, so that the refrigerator look clean and organized. I don't know your all right. So another one is the open water bath, sometimes used in the tissue culture laboratory to preheat medium, are a major source of contaminations as well, because you need to preheat. For example, if you culture the mammalian cell line, yeah, and your media usually you store at 4 degrees Celsius. So you need to preheat it to 37 degrees Celsius before you can culture your cell. Okay, usually you put in a water bath, and this water bath can be very dirty if you did not change the water or you did not put any disinfectant. Yeah, because everybody use the water bath and you do not know how they handle it, right? So it is very important that for you to make sure that the water bath that you use to heat up, okay, or preheat your medium, clean, and you put the disinfectants inside to make sure that there's no contamination to your cell that you want to culture. And the water used for humidity inside the incubator should also contain a labor germicide, and surfaces should be wet down with the germicide frequently. The tray that are removed from the incubator between the experiments should be decontaminated or autoclaved and stored in a clean place until the next use. Okay, and always clean up any spill medium immediately, since the media is the very best way to grow unwanted bugs, balls, or whatever it is, and any other not readily identifiable identity in the culture, because it's rich of medium. Yeah, the medium are rich of nutrients and anything can grow on it. So one, it is any spillage, you need to clean it immediately, and also minimize the movements of the people in and out of the culture, and needless to say, no food or drink in the culture room. Okay, for your information, during my times of study in undergrad, I noticed that some of my classmates, they party inside the the lab. They use the beaker to cook their Maggie mee, which is very dangerous, and to me is very irresponsible. So never ever do that in the lab, because you can swallow poisons, you can swallow chemicals which may kill you, right? So never ever take any food or drink in the cultural room. If you want to bring your food, bring it outside. There should be a space for you to store your food or drinks. You have to do it outside of the lab, not inside the lab. Okay. So what is the aseptic techniques? Okay, it's basically a combination of the procedures designed to reduce the probability of infection and contamination. This is extremely important because you don't want your cell line to be contaminated. So proper aseptic technique must be practiced from time to time to assure that your culture are clean from contamination. So the elements of the aseptic environments, which includes the work phase, the personal hygiene, the reagents, the media, and also the culture. This is very general. Okay, what is the work surface? I think I do not need to elaborate more. Yeah, and then the sterile handling, you probably need the swabbing, the capping, the flaming, handling bottles and flasks, pipetting and pouring. These are the proper sterile handling techniques that you need to master in order to reduce the contaminations. So what is swabbing? Basically, it swab down the work surface before and during the work, but particularly following any spillage, or swipe it down again when you finish. So swipe before, in between, and also after you finish it to make sure that the work surface is clean for the next use. Okay, always practice this. And also the capping, the screw cap should be covered with the aluminum foil to protect the neck of the bottle from sedimentary dust. Because if you don't wrap it with the aluminum foil, when you open it, the dust can get into your medium easily and contaminated your medium. And flaming, because you use a lot of this technique. Okay, so when you working on the open bench, so the neck of the bottle or the screw cap before and after opening the bottle, after closing it, so you need to flame a bit. So example, the pipettes, the glass should also be flamed before use, but not the plastic pipettes, yeah, because it will get melted later. The work close to the flames, and then screw cap should be placed with the open side down on the clean surface and flame before replacing it on the bottom. You don't put it upside because the dust can trap, okay, can get into the cap. So you put it up down, okay, the open side down. And before you put it on back to the bottle, then you flame it again, right, to create a sterile environment. And then when working on the bench, the bottle should also be kept vertically when opened and keep them on an angle and shallow as possible. And the culture flap should also be let down horizontally when open, held at the angle during manipulation, because you want to reduce the possibility of the dust or the other contaminant getting to your media. Pipetting is also necessary to insert the cotton plug in the top of the glass pipette before sterilizing to keep the pipette sterile during use. So the glass pipette, especially when you wanted to sterilize them, so you need to put a cotton plug on the neck, okay, so that when you heat it up, like to 170 or 180 degrees Celsius for like 6 hours. Then when you take it out, the internal glass environment is totally clean and sterile. Okay, so when you use it, then you may remove the cotton plug, or you can just put it right into the pipette gun and then aspirate your media. Pouring also can be a very the wrong pouring technique can introduce contaminants as well. So whenever possible, do not pour from one sterile container into another unless it is to deliver all the content into one single delivery. Yeah, because the more you pour in, the more frequently you pour into different small jar or small vials, then the chances for the contaminant to get into the media are very high. So if you want to pour, make sure it's one-time pouring, okay, in a single delivery. This is the aseptic techniques that I capture from videos. You can just read on, okay, and I am going to show you a video. Yeah. Video. Can you hear the sound? You get the audio?
No.
Okay.
It is
Slow,
soft.
slow.
Oh. No sound.
It
There's a
is
sound,
soft.
but slow.
Very slow. Okay. If that's the case, then you go to YouTube, okay, and you go to aseptic, you tap the aseptic techniques and look for your BioRed laboratory, because BioRed is a very good brand as well. Very good brands, and they provide lots of good video for aseptic techniques, and there are also few aseptic techniques here you can look at. Okay, so also in the course here, there are 2 videos that are embedded that you can also look, have a look on it. Okay, boleh tak?
Boleh, Doktor.
Okay.
Boleh, Doktor.
Because this lecture is quite short actually, because aseptic technique is very general, it's quite simple, but for you to practice it, there's a challenge, as I said. Okay, it look very easy. Okay,
Recording
easy.
stopped.
But to practice it, to make it a consistent, make it a good habit, that's quite challenge. Like you keep your refrigerator clean. That's a challenge, isn't it?
Yes.
You look at your house, because you are not living alone. You are living with your family member. Everybody have their own style, right? Have their own attitude.
Yes.
Yeah. Some people just chuck everything in.
Yeah.
Yeah, they get the fish, they get the meat, or what, the chicken, and they just put in. They don't care. They don't put in a proper container and then they freeze it properly. Correct?
Ya,
Ada
Doktor.
ada yang berlaku tak dekat rumah?
Ya, ada.
Sometimes you just fight kan kerana bau. Yeah. So that's why lah, in the lab pun sama, because you are not the only one that use the lab. Your colleagues, your other classmate probably use the lab as well, but you know they have different kinds of attitude and mindset. So be thoughtful when you go to the lab and practice good hygiene, especially for those if you wanted to do FYP with me. Make sure the lab is clean, okay, especially if you do some molecular work or tissue culture, okay. And plant tissue culture, I think Dr. Ho will teach you how to. Now, probably slightly different of the aseptic technique for plant because animal and plant are slightly different, of course. All right. So, siapa lagi nak scan? Ada tak? Nak scan attendant?
Yes, doctor, I haven't scanned.
Haven't scan, ah?